So we worked in collaboration with a company called the Electrospinning Company, and what we've developed are effectively like we call them sort of mini scaffolds, but I guess you could describe them as like children's like shreddies in terms of breakfast cereal. So what they're actually composed of are these structures, random fibres of scaffold material, and they're from the hexagonal shaped, and they range from sort of 100 micron to 1,000 micron in kind of diameter, but they're only 50 micron thick. And what we're able to do is we're able to seed those with cells, and the cells use the scaffold as a platform upon which to adhere, and upon which to grow, and upon which to proliferate. Yeah. So there's a couple of things that we've done with the material, and one of the things that we've done with the material is to incorporate iron into it. And that doesn't affect the cells, but what it does is it makes the scaffolds attractive to a magnet. So what we're able to do is we're able to use magnetism both in culture in a well plate as well as in a tube in order to move around the scaffolds, and by doing so we move around the cells. So what that means is that there's better diffusion of nutrients into and out of the cells, but it also means that it's practically easier to be able to dispense them into well plates and to be able to culture them and to be able to use them. I think, in cutting techniques, what we do is we culture things in 2 dimensions on a plastic strata, which is completely unnatural. But we've been doing that for the last 40 or 50 years simply because that's all we know. Yeah. I think we can see an increase in the number of publications referring to 3-dimensional effects of cells when they're maintained in 3 dimensions. And I think what we'll see is a plethora of more papers and posters coming out, which will demonstrate that actually cells in 3D are a lot more physiological than those ones in 2D. Yeah. And so what we like to end up with, being a contract research organisation, is providing people with screens that are more physiological. More physiological is in the sense of they're maybe co-cultures of cells, they're in 3 dimensions, and that means that they're more representative of the normal physiological state.